compare our method with the other three partitioning methods. So first, we want to get an idea the performance of our model with and without SIF constraint. So here we use another method, SIF, no SIF. Basically, it has the same structure as SIF, except that it does not have the SIF subnetwork. And then we also compare our method with the conventional nighttime method and daytime method. And to evaluate these different partitioning methods, we use both synthetic data and also real in situ measurements. The reason we start from synthetic data is because in reality, we don't know the truth of GPP and respiration. So we run the scope model at Harvard Forest during its growing season, and to get the synthetic data to test if our model can be normally functioning under a simplified setting. And then we move to the field observations from 3 different sites, including Harvard Forest and a wheat site in Avignon in France, and also a rice paddy site in South Korea. So one thing is that for this wheat site, it is Mediterranean. So it is seasonally water limited, which means it provides us a nice opportunity to test our model under water stress. So here, this is a result showing our model's performance when it is tested against the scope simulation. And here, the left panel shows the comparison between scope GPP and the GPP partitioned by our method. It shows a high agreement with a high R square, but we think the high R square of GPP is not sufficient to prove the model performance because the R square can be indeed driven by the strong diurnal cycle and also seasonal cycle of radiation. So we normalize GPP by the radiation to get the light use efficiency, and also we find good agreement between the reference scope value and our partitioning results. And then we also further take a look at whether our model can learn the functional relationship as implemented in the scope model. So we check 2 relationships. The first one is GPP PAR relationship, and also the light use efficiency VPD relationship. And we find our model can learn these relationships, reproduce these relationships. So this is the results for our model. And then we did the same thing for the other 3 methods. So generally speaking, all methods, they can do a reasonably okay job in reproducing GPP and its associated functional relationships. But for ecosystem respiration, this is not the story. So here, the upper panel shows the comparison between scope respiration and the respiration predicted by our model. And you can see there is a okay performance for our results. And then we remove the SIF subnetwork and find that for the no SIF, the predicted, so here you can see that the scatters, they kind of cluster above the diagonal line, which means that the estimated or the partitioned respiration by no SIF, it kind of underestimates the respiration, in particularly for the scatters with more red color, it means under higher temperature. And this underestimation issue can be more visible if we plot the relationship between air temperature and ecosystem respiration. You can see that for an no SIF, it shows limited skews in reproducing this relationship when the temperature is outside the nighttime regime. And then we further check for nighttime method and daytime method. And for the nighttime method, we find similarly underestimation issue under high temperature, and also for the daytime method, generally it is able to reproduce the relationship between air temperature and respiration, but it shows larger deviation from the reference relationship throughout the entire air temperature range. So you may wonder why this would happen. And the quick answer is regarding the different distributions between day and night. So here, if we check the scope simulation, for the temperature, including canopy temperature and soil temperature, they show quite different distributions between daytime and nighttime. Because temperatures, they are major drivers for ecosystem respiration. These different regimes can further lead to a different distributions of ecosystem respiration between daytime and nighttime. So basically, for the daytime ecosystem respiration, it has overall higher magnitudes and also steeper slope with respect to air temperature. But for nighttime and daytime methods, they are assuming that the respiration-related parameters remain invariant through day to night. So they either fit the parameters using nighttime data and then extrapolate to daytime, or they do it conversely, fit the parameter using daytime data and then extrapolate to nighttime. So here the result means that this extrapolation strategy may not work well when there are higher discrepancies between daytime and nighttime conditions. And for the NNO-SIF method, because it is only constrained by NEE, and this only constraint shows limited skews for daytime flux partitioning. But for our method, because it is additionally constrained by SIF, so for when processing the daytime samples, GPP is directly constrained by GPP, as shown by the red dashed line here. And this constraint can further propagate to the ecosystem respiration. This is because when GPP varies, the respiration should also correspondingly covary because they need to ensure their difference still, like NEE, still match the observed NEE. So it means that the SIF actually can also constrain ecosystem respiration, but through a more indirect way. But this indirect role would be very critical when night conditions is different from daytime conditions. So those are the results regarding synthetic data, and now we can move to more about field data. So here, when we find that one benefit of our approach is that when it is applied to the rice paddy site, it shows better performance in capturing the instantaneous effect of harvest on GPP. So here, the rice paddy is harvested at the day of year of 248. And after the harvest, the observed SIF, as shown by the green triangles, it shows a sharp drop. And informed by this SIF information, the GPP predicted by an SIF, it shows it is more sensitive to this sharp drop. And we think that this better performance is not just because we include SIF here, it also related to how we include it. So here, SIF is included as an extra module in our machine learning algorithm, which means that it can directly join the optimization of our model, and it can directly inform the model parameters. And we also take a quick test by simply including SIF as a predictor for GPP, but we find less improvements in capturing those more subtle changes within short time period. And another thing I wanted to mention is regarding water stress. So here the Avinia site, it is a seasonally water limited site. So in the records captured by eddy covariance data, there is a 20-day water stress period, as indicated by those dark points here. So the points, they are colored by evaporative fraction. It is a proxy for soil moisture because for this site, we do not have available soil moisture measurements. And under those conditions, we find for an SIF and an no SIF, they are able to capture a moisture limitation effect on ecosystem respiration. This can be seen by the relationship between air temperature and respiration. So basically it first increases and then it transitions to a decreasing relationship when it is under low evaporative fraction conditions. But for the nighttime and daytime methods, they do not include moisture as an direct input to the method. Instead, they are using moving windows to indirectly account for moisture effects. So in nighttime method, so they just keep increasing with the temperature because in the equation, it is defined as an increasing trend. But here, we also notice that for the daytime method, the overall magnitude under those water stressed regime, it is relatively low. So as we mentioned earlier, our goals are not limited to just partition fluxes. We are also interested in retrieving the relationship between GPP and SIF. And this plot shows the relationship, those retrieved relationships. So basically the x-axis and y-axis, they are all predicted by our model. And here for the Avenian side and also the South Paddy side, the retrieved relationship is more linear. But for the Harvard forest, it is more nonlinear. So this also supports that our model can retrieve both linear and nonlinear relationships. And because here we are estimating SIF as a function of GPP plus environmental inputs, it means that by learning this function, we can also learn how the relationship between GPP and SIF are associated with changing environmental conditions. And the first point we find here is regarding atmospheric dryness. Here we find that the slope of GPP versus SIF, they tend to be smaller when the air temperature, when the atmospheric dryness becomes higher. So basically the scatters are colored by the VPD, vapor pressure deficit level. We find the slopes become smaller for those more red points. Also, this holds true for an extreme drought experiment we conducted using scope simulation. And this dependence on humidity is associated with stomatal behaviors. So under drier atmosphere conditions, the plant stomatal, they turns to close to avoid more water loss. And this further affects the let use efficiency and decrease GPP. But in the meantime, the SIF yield is more insensitive to the stomatal behaviors. So, and it shows a more moderate change for SIF to air dryness. So, and this is the reason why the ratio becomes smaller under drier condition. And here our model results showing that it can learn this dependence based on a data-driven approach. And in addition to atmospheric dryness, we also find dependence of GPP-SIF relationship with respect to illumination conditions. So here we fit the relationship respectively for cloudy conditions as shown by the blue points and also sunny conditions. And we find they have different slopes. So basically the slopes becomes higher under cloudy conditions. And this is because on cloudy days, the light can be penetrated into deeper canopy levels because of more diffuse radiation ratio. And this allows for higher light use efficiency and increase the overall canopy GPP. So in summary, here we develop a framework which can first partition NEE and also retrieve the relationship between GPP and SIF. And also we observe better performance under high temperature and also when human disturbances is affecting carbon fluxes. And also another byproduct is that it can review how the GPP-SIF relationship responds to physiological factors. And this learned knowledge potentially, if we apply this framework to satellite data, it can hold the potential to achieve better estimates of global scale GPP estimates. So those are the results we have done before. And I also would like to briefly mention two ongoing works I'm engaged in regarding carbon flux partitioning. And the first one is led by a collaborator, Kai. He is at the University of Valencia. And he is trying to develop a unified protocol to compare different partitioning methods. So because there are more and more partitioning methods coming up recently, but those methods, they are evaluated by different teams. They are not under a unified, like evaluated using a unified benchmark data set or a protocol. So they are trying to build up a benchmark data set to evaluate different partitioning methods to give it a more transparent assessments on the weakness and also advantages of different methods. So he will be presenting this work in the upcoming EGU conference, and you are very welcome to drop by his talk. And another work I'm collaborating also with Kai is regarding a hybrid modeling framework. So basically, we are trying to combine both machine learning and also some process understanding, for example, the Madeline model, to jointly partition NEE and evapotranspiration. And it's kind of like an extension of this work, and also another ET partitioning work done by our group a few years ago. And so you are also very welcome to reach out if you are interested in it, and we hope we can share more updates later. So these are the brief overview of our previous work and also ongoing work regarding carbon flux partitioning. And feel free to let me know if you have any questions. And also, I really appreciate that there are so many people coming to listen to this talk. In particular, there are some people from different time zones, like the very late nights in Asia and also in Europe. Yeah, so feel free to ask any questions if you have right now. Yeah.
I'll just say, first of all, thank you so much for your presentation. If you like, there are a couple of questions already in the chat section if you'd like to start.
Yeah,
for
I saw
example,
those.
you see them?
Yes, I saw it. So the first question is whether the validation is based on the half-hourly data set. So here for the training, validation, and test, we are all conduct them at the half-hourly or hourly data set. It depends on whether the data is available at hourly or half-hourly scale. Yeah. And another question is, how do we distinguish sunny and cloudy conditions? So we distinguish it based on the ratio of diffuse radiation. So if the average radiation, diffuse radiation ratio is above 50%, then we think it is, we evaluate it as a cloudy day. Otherwise, it's a sunny day. And the third question is about whether the neural networks were trained on time series directly or aggregated on some form. So maybe we can get a better sense of this, how data look like when we check the code later. But for now, we are training the network using half-hourly or hourly time series, but we shuffle those hourly time series into when we divide them into training and validation test sets. So the short answer is it is trained on half-hourly time series, but not on a sequential order of different, like from first day to the last day. Yeah. And there is another question about more about scope. And can you explain more about scope, and can the simulation results from scope be considered as a true value? Yeah, so scope is a more process-based model. It can simulate plant photosynthesis and also the solar-induced fluorescence, and also it considers the radiative transfer process within the canopy. The reason we choose scope here is because we need some known truths that includes both carbon fluxes and SIF, because our goal is to include SIF here, and scope is more widely used in the community. And it's a great question to see if it can be considered as true value. I would say it's more like a simplified version of the complex interactions within an ecosystem. So if we use it as a baseline, so if the model cannot work even in a simplified setting, then we would not be too optimistic in extrapolating this method to field observation. And also before we using the scope simulation, we also compare our results, scope simulation with the real measurements from Harvard Forest, and we find acceptable agreement between simulation and real measurements. And another question is that the method has been tested on deciduous forest and water-limited croplands. Do you expect to test the method in other ecosystems under different climatic conditions in the future? Yes, I really want to do it. The reason we are limited to these 3 sites is because at that moment, during the collaboration, the available SIF data we can collect are major from this. We only have these 3 sites. But for sure, I want to apply it across more wide climate regimes, because here it's hard to detect. I really want to test the performance and also the discrepancies between different partitioning methods under extreme climate conditions, but it's kind of tricky to find those conditions for Harvard Forest or for the rice paddy site. So I would be very curious about whether the method can robustly examined under like soil, under like high temperature or under compound drought events. Yeah, so that can be a very promising plan for the future. And another question is wondering, how do you get SIF values? Did you measure from the eddy covariance tower or from remote sensing product? So here the SIF is collected by, so again, thanks to my collaborators, they build up the site level SIF measurements. I'm not the expert on how to measure SIF, but they build separate SIF measurements in companion with the eddy covariance tower. So it is more like in situ measurements. So it has higher temporal resolution, like half hourly or hourly. But this also is the limitation of our work. So it means we need to use hourly or half hourly SIF measurements for now. But if in the future we do plan to use the remote sensing data from SIF so that we can expand the size to test from the size with SIF measurements to to the most of the size distributed globally. So we do have the plan to use SIF, remote sensing SIF, but it has smaller temporal frequency. So it remains unclear under that condition to what extent the SIF information can inform the carbon flux partitioning. And another question is, the scope simulations for NEE and GPP are better than tower estimates. So here, for the real measurements, it's hard to see what is the truth value for GPP. And for NEE, we would think the tower measured, the NEE measured by tower would be the ground truth. So ideally, we hope our scope simulations NEE to be as close as possible to the NEE collected from eddy covariance tower. But for GPP, when we compare scope simulation with tower-based partition GPP, we find okay performance, generally acceptable agreement between scope simulation and partition GPP. We think this is acceptable because even we just check about the tower estimates. If we use different partitioning methods like daytime versus nighttime, we still can observe quite different results just stemming from different partitioning methods. So here, we would not expect very high agreement for GPP between scope simulation and tower estimates. And another question is how long have to be the time series you use to train your neural network method? Does it work with conventional NED ID covariance measurements? Yes. So the time series here for Harvard Forest is 2 years, and for the Avenian side it is 1 year. For the South Paddy side is also 1 year. So the kind of limited temporal coverage is limited by the availability of continuous SIF measurements. So I would expect our work, so our model will be workable with conventional ID covariance measurements as long as it provides at least 1 year data. And also, if we are not limited to stack level SIF observation, we are using remote sensing SIF products, then we can apply our framework to the FluxNet site. Yeah, so I think, yeah, so those are the questions. Oh, I see
Maybe if you want, you can maybe take this one last question and then we can move on to the code demonstration.
Can I jump in?
Yeah.
Okay, sorry. I couldn't type really long because my question is maybe it's not clear to me even. So Vivi, that was fantastic. So can you come back to your figure related to evaporative fraction, ecosystem restoration? Yeah, that one. So yeah. I'm just wondering for the model that includes SIF and not non-SIF, they looked similar trend, which was, kind of great to catch the water stress. But I'm just wondering for the ANN model, for the model with no SIF, supposed to be kind of similar to the nighttime restoration, which the previous figure, when you compare to the scope with nighttime restoration and scope with this model, looked similar. I remember for the high temperature, the models with the no SIF couldn't pick the trend, right? This figure, yeah. So I'm just wondering how possible with no SIF captured that trend really good, while it behaves here similar to nighttime respiration partitioning. Is it clear?
Yeah. Okay. Thank you for this fantastic question. I think you pointed 2 different points regarding how they estimate respiration. So regarding this moisture effect, here the reason why a no SIF can do a better performance is because it's not mostly because it uses a more combinations of different inputs, including moisture, to directly consider its effect on ecosystem respiration. So here I would say this shape is not mostly driven by the inclusion of SIF. But we do observe that for the no SIF, it shows some abnormally high ecosystem respiration values as shown in this circle. So it means that adding SIF here, it further constrains the respiration estimates to be within a reasonable range for this part. And here, regarding the temperature point you mentioned, I would say this is less relevant to the moisture dependence. And because these 2 figures, they are tested against the different data, different climate conditions, and this is more relevant to the function for temperature. So the reason why a no SIF looks like a nighttime method here is because both of them they are kind of fit a function using nighttime data and then extrapolate to daytime. So just imagine here, if there is no SIF subnetwork, for the unknown SIF, it will be look like you feed a response function using nighttime NEE, and then when GPP is 0, and then when you came across daytime data, you extrapolate to daytime. So that's the reason why they two look similar. But here they two does not look similar is because when they fit this nighttime respiration, like respiration function, nighttime only use temperature as driver, but most neural networks, they use more inclusions of different predictors. That's kind of different. There are different aspects regarding how these two.
Okay, thank you, Weiwei. Just a question. Did you say this figure only for the nighttime respiration?
Oh, sorry, my typo. No. This is for the entire day. I just yeah.
So here,
sorry, maybe we can continue this conversation after the.
Thank you, Weiwei.
Very interesting,
no problem. Yeah, thank you for those fantastic questions. And then right now I will jump very quickly to the code sharing part. But before that, I would say if you are interested in running the model, the code, but you don't want to install local environments from your own code, your own machine, you can just click this launch binder, and then it will build up a environment I use to run my code from your end. Yeah. But here, right now, I will just show the codes from my local lab. Sorry, where is my? So yeah, so here, can you see my codes right now? Okay, cool. So here I will just very briefly go through the example we used our method for the scope simulation, and there is another one used for Harvard Forest. Basically, they are similar processes. And here you can find there are 4 key components. So the first, we import data and pre-process it, and then we train the model. And then we use the model to predict carbon fluxes, and finally we evaluate the model. So to pre-process this data, we first import the data as data NN, and those are the variables we need to train and evaluate the model, basically outputs, inputs, and truth value for GPP and ecosystem respiration. And then we divide it into training and test sets. Oh, so here, back to the question someone mentioned earlier, it looks like this. It's a time series. And then we randomly divide it into training and test. So here, the temporal sequence is no longer hold. And then we select model inputs, basically predictors for GPP, respiration, and SIF for training and test size. And we also select model outputs for training and test size. And then we do a standardization for the model inputs to make them have the 0 mean and a variance of 1 to facilitate training. And we similarly also normalize our model outputs, NEE and SIF, by the maximum value of their absolute values, because then the absolute magnitude will be scaled to 0 to 1. This will enable fast convergence of the model. And regarding the second part, here, by training the model, here you can see there are 3 components to train the model. The first function, the first function is get prediction model. So it is used to define this general structure of our model. You can see that there are 3 subnetworks. The first one is GPP, and then respiration and SIF. And for GPP, we first define an input layer that receives climate and biotic factors, and then we define 2 hidden layers, and then we first predict the log value, and then take the exponential value to make sure GPP is not negative. And then we multiply it by a par label to constrain nighttime GPP to be 0. And then this is the subnetwork for respiration. It follows a similar structure, and we further take the difference between respiration and GPP to get the NEE estimates. And here, from this point, we are adding the SIF subnetwork. So we first define an input layer to receive environmental inputs that controls the GPP-SIF relationship, and then we combine this environmental inputs with the model estimated GPP, and then finally we also similarly add hidden layers, and finally we get the SIF prediction here. And in the end we define our model by specifying their inputs and their outputs so that these three networks can be combined together. So this is the purpose of our first function. But it's not sufficient to just define a model, because in this way, we can only get model estimates. We do not know how to train this model. So right now, we need a loss function to link our model estimates to real observations. And this is here, because here we are using two model outputs, and it's tricky to how to balance these two components. We want to decrease both. So here we use a custom layer to achieve the multitask learning. So basically we want to minimize both the loss for NEE and also the loss for SIF, but the weights for these two terms, they are weighted by the uncertainty of the data. So basically we first define some trainable parameters, the log of variance, and then we take the exponential of this log as a precision. So this is a weight for NEE and for SIF. If NEE has lower uncertainty or lower sigma, then it will have higher weight. So we assign higher weights to some variable we trust more. So basically this loss function achieves this multitask learning by here. And we finally add this loss to the model structure. So the third part, get trainable model, is working to combine our defined model structure with the loss function. So here we similarly define the input for this model, and then by using the prediction model function, we can get the predictions for NE and SIF. And then in the meantime, we also know the truth value from NEE and SIF. So right now we use the custom loss function to link these two together to get the final output. This is a loss. So this quantifies the distance between our model prediction to the real observation. And this cell shows how we jointly use this above three components to define and train our model. So first we define our model structure, and then we define the model to be trained, and we compile the model, and finally we fit the model. And from this point, we will already get a well-trained model. And then we want to retrieve the outputs or the estimates, like including NEE, SIF, GPP, and respiration from the well-trained model. So here we also define two functions. The first one, get layer output, obtains the intermediate layer output from our model. And then we use another function, flux SIF predict, to finally retrieve these four variables we are interested in. And after we get the predicted model, the values, we start to evaluate the model predictions by comparing the time series of our partition GPP versus the GPP simulated by scope. So this is what I run like this morning, and it works good to reproduce this scope simulation. And also, another thing we are interested in is the retrieved GPP-SIF relationship. So here we plotted the leftmost panel shows the relationship originally implemented in scope model. So both x-axis and y-axis, they are simulated by scope. And the middle panel shows the relationship derived by our method. So it is both the SIF and GPP predicted by an SIF. And the rightmost panel shows relationship between partitioned GPP versus the scope SIF. So basically for all three of them, you can see a quite similar relationship. So it further verifies that our method is able to retrieve the GPP SIF relationship. Sorry, I realized I didn't run my model right now, but if you are interested in and get a sense of how long it will take, I can run it right now. It is very fast. So basically, the most time-consuming part is this part, but it can be done very quickly. And we get the prediction from this cell. And finally, we plot the time series. So here you can see the results is consistent with the previous one. And finally, we also get the GPP SIF relationship. So this is what we do for scope simulation. And another example is for Harvard Forest. You will see a very similar example here, but based on the provided data for Harvard Forest measurements. So yeah, so you are very welcome to run this model from your end using the binder platform. Yeah, so this will be my very brief introduction to the code part. And feel free to jump in right now if you.
And we got 2 questions in the chat
Yeah.
about code.
How were the hyperparameters tuned? Did it take a long time? Yes, great question. So here in this notebook, we do not show how we tune the hyperparameters. I just put the final choice of our hyperparameters, like basically the number of neurons for each hidden layer. And before that, we do a hyperparameter tuning using the Sharper library. So basically, we go through different combinations of hidden layers. I think for sure it takes longer time because it involves multiple runs of the algorithm. But I would say, if at the start point, if I just randomly choose normally use the hyperparameter setting, and then my baseline model, it works okay at that moment. So it means that the hyperparameter tuning, it makes your model better, but it can, but before the parameter tuning, it's already a workable model. So it does not affect too much to my results. And another question regarding to the outliers. Regarding the outlier, I'm not very clear about the outlier regarding NED or regarding SIF, but so here we for the NED and SIF observations, we should simply use the original observation without removing the outliers. But if it is flagged as low quality, then we remove it. So basically we only keep data points flagged as good measurement quality, and we do not use the gap field NED in our case, because it will be a circular, because the gap field is based on the learned relationship between carbon fluxes and climate inputs. And here, what we are doing is to learn this relationship. So in short, we just use original measurements and remove the measurements, the bad measurements because of the measurement quality.
Yeah, big thank you to our speaker today for such an excellent talk. And if anyone still have questions, you can write to Weiwei. And thank you all for the engagement and thoughtful question today. And see you all in our future events. Bye.
Thank you so much, everyone.
Bye. Thank you again very, very much. Very interesting.
Thank you so much.
And I'll email you with some questions.
No problem.